So hello everybody. Today we are going to understand the second chapter of ICSE examination biology for class 9 and 10. The name of the chapter is Structure of Chromosome, Cell Cycle and Cell Division. So first, to understand this, we need to focus on what is chromatin. So chromatin is a thread-like structure which is present inside the nucleus of the cell. Let us consider a plant cell that has a cell wall, that is a non-living structure made up of cellulose that we have learned earlier, right? So inside the cell there is a nucleus, and the nucleus contains thread-like structure which is known as the chromatin. Chromatin is made up of DNA and protein. A special kind of protein is present in DNA. So we will learn as we further proceed. Okay, so this chromatin is present. It is visible by some special kind of staining procedure. Especially it is visible during cell division, because during cell division this chromatin condensed and formed chromosome. It is chromatin fiber. And during cell division, this condensed and formed chromosome. Fine. So this chromosome was first discovered by Welder Fleming. Fine. So we will be now shifting our focus on chromosome. As our chapter revolves and our syllabus include the cell division process, so we will be learning the cell division basis on the chromosome. The chromosome was first discovered by Welder Fleming, as I told, and he observed cell division in the mitosis process. There are two kinds of cell divisions. One is meiosis, other is mitosis. So Welder Fleming observed cell division in mitosis. Fine. So the chromatin is made up of DNA and proteins, and the name of that protein is histone protein. About 40% of our chromatin contain DNA, and the rest 60% is protein, that is the histone protein. DNA is a double helical structure, that is it has two helix. This is DNA, and the protein histone stays in octamer condition, that is in a group of 8. Fine. It's DNA and it histone. We will be first learning about DNA. DNA stands for deoxyribonucleic acid, and it is made up of repeated groups of nucleotides. And what is nucleotide? Nucleotide is actually, it contains three things. One is pentose sugar, then it contains a phosphate group and a nitrogenous base. Repeated nucleotides forms DNA strand. It is these two strands are complementary. The structure of DNA, this double helical structure, was mainly studied by Rosalind Franklin, but Watson and Crick got Nobel Prize for this double helical structure, ladder-like shape, which has a ladder-like shape, and for this structure Watson and Crick has got Nobel Prize in the year 1962. Fine. I told that DNA is made up of repeated structure of nucleotides. What's nucleotides? Let's focus on that. this nucleotides is made up of three things: a phosphate group, a pentose sugar, and a nitrogenous base. Fine. And the structure of nucleotides looks like it is known as pentose sugar because it has 5 end, or we can say 5 carbon. So in some organism, the DNA is not present. Instead of DNA, RNA is present, and the RNA stands for ribonucleic acid. So this RNA also contains this repeated structure of nucleotides. But the main difference between RNA and DNA is that the nitrogenous bases present in DNA are A, T, G and C. A stands for adenine, T stands for thymine, G stands for guanine and C stands for cytosine. While in case of RNA, the nitrogenous bases are A, U, G, C. That is, adenine, guanine and cytosines are similar, but U is uracil. That is, thymine has been replaced by uracil in RNA. Fine. This A and G are purines, and T and C are pyrimidine. Also, A and G are purines and U and C are pyrimidine. So there are three pyrimidines: A, C and T, C and U, and two purine, that is A and G. That this is repeated nucleotides, right? So this nucleotide contain a pentose sugar, a phosphate group and a nitrogenous base. As I told that it's complementary to each other, the two strands are complementary to each other. That means they have complementary bases to be paired. And this is a strand and this is the other strand. They are joined by hydrogen bond. These are the hydrogen bonds. This is one nitrogenous base. This is the other nitrogenous base of this strand, and they are joined by hydrogen bond. So as we know, as we are considering in the case of DNA mainly, so if this is A, the base pairing happens in the pattern like A base pairs with T by a double hydrogen bond, and G makes 3 hydrogen bond with C. Similarly in RNA, A make 2 hydrogen bonds with U, but we will not consider this as its DNA and not RNA. So as it's A, it will be pairing up with T and 2 hydrogen bond will be formed in between. Let us consider this is G, so this will be C and there will be 3 hydrogen bond. So if this becomes this, that means if this is T, here will be 2 hydrogen bond and this will be A. Correct. Because A bonds up with T and G bonds up with C. I think it's clear. Okay. Thus, moving forward, let us now number these carbons of the pentose sugar. Here stands 1, 2, 3, 4, 5. Thus, we could say that the phosphate group linked to the first carbon of a pentose sugar of a nucleotide is linked to the fifth carbon of the pentose sugar of the previous nucleotide, and the nitrogenous base is linked with the third carbon of each nucleotide's pentose sugar of each nucleotide. Fine. Thus, let's move on to the next component of the chromatin, that is the histone proteins. So, histone is a kind of protein. Histones remains in an octameric condition. Octameric condition means they remains in a group of 8. 1, 2, 3, 4, 5, 6, 7, 8. That is in a group of 8 they remains. Fine. The DNA strand which we have learned previously just now, each histone octamer is wrapped around by 2.5 turns of DNA, and this structure is known as nucleosome. Nucleosome is the basic structural and functional unit which makes up the chromatin. Fine. That is, repeated nucleosomes makes chromatin. Thus, repeated nucleosome makes chromosome. So, as we have learned the things which makes up the chromosomes, thus this part ends here. And let me remember you one thing, that these DNA remains in a too much coiled condition, like the wire of a telephone. Previous days telephone, the wire is too much coiled. This DNA also, this chromosome, this full chromatin fiber remains in that coiled condition, which coils around itself only. Because almost 2 meters of chromatin is present in one single cell. That means it's a huge length compressed or adjusted in a very minute cell. Thus, they need to be compact, so they coils around itself. Fine. So this part ends here. Now, in the next part, I will be discussing about the cell cycle and cell division. Thank you for watching, and please support me and subscribe my channel. Also share my video with your juniors and fellow mates so that they could also get some help from my videos. Thank you.